Hello everyone, welcome to week 6. Hopefully you had a decent break. This week we'll be covering immunity and physiology. And physiology is a big chunk. Immunity is it's a three-part lecture. You get 2 of them in week 6, and I think the other one in week 11 or something along those lines. And if you've done Bio 3-4 in BCE, it does help. And if you did the Bio Bridging course, I'm pretty sure you went through it as well. So it's kind of like Bio 3-4 immunity, but with a bit more in-depth and just doesn't cover some of the things that you would in Bio. So starting off, this is just the immune system and immunology. Right, so innate response to pathogens. So there's obviously innate versus adaptive immunity. Hopefully you guys know that because you would have come across it at least once if you did the bio course, bio bridging course, or the bio 3-4. But essentially innate and adaptive are different, obviously. And innate have basically a broad spectrum, and it's basically the same for any attack or pathogen. Whereas adaptive is very highly specific, using B and T cell receptors. And innate is fast to act on, and also short in duration, whereas adaptive response is slow to work on, but also long in terms of duration. And other ones, they're not that important, but it's just there for your reference. Okay, so lines of defense. So there's basically 3 lines of defense that we normally talk about when we talk about immunity. First one is the physical barriers. They're non-specific. And then there's the innate immunity, which is second line, which is also non-specific. And then there's a third line, which is adaptive immunity, which is specific. So first line, this is pretty straightforward, so I won't go into too much detail. You guys can have a read over it later. But basically, there's just different types of physical barriers that are preventing pathogens and things that are meant to be in your body getting into your body. So just have a read through them. It's basically, they all act to block them out, disarm them, or flush them away. So basically just trying to prevent any of them being part of the body or being in your body. Okay, so cells of immune system. So this is how immune systems kind of come apart. There's immune cells or immune cells are also called white blood cells or leukocytes, more commonly referred as. And they're all derived from polypotent stem cells in the bone marrow. We'll go through some of these ones, especially highlighted in green, which is part of the innate immune system a bit later on. So here it is, innate cells. So there's 3 different types. There's phagocytes, there's cytotoxic and inflammatory cells. And basically they all have different functions. So phagocytes, this is a group. So there's neutrophil, monocyte, and macrophages. They all basically engulf and consume pathogens via phagocytosis. Neutrophils are the most abundant. That's one of the most important facts. Monocytes are monocytes when they're in blood. And if these monocytes move into tissue, they are called macrophages. So they're the same thing, depending on where they are present. As you can see, monocytes are also APCs, antigen-presenting cells, which we'll go into a bit later on, and also macrophages are as well. Other ones you can read through. Again, it's not really important to kind of like memorize all the different descriptions of these cells, more so to understand the overall basic function and integrate it with the immune system in general. So now we want to go talk about the cytotoxic cells. These are the natural killer cells and eosinophils, I think that's how you pronounce them. These release molecules that kill foreign or infected cells. So whereas phagocytose, which we'll explain a bit later, basically engulfing the pathogen, these cytotoxic cells, they would release molecules, harmful, things like peripherin, that will kill foreign or infected cells. So they don't need to engulf anything. That's the cytotoxic cells. And then there's the inflammatory cells, which basically release histamine, causing inflammation. Inflammatory is actually not mentioned in the lectures, but that's how I learned it in Bio 3-4. So I just wrote that down. But yeah, just keep that in mind. It's not technically in your mid lectures. But basically, it's still the basophil and mast cells are still the same type of cell. They basically release histamines and basically cause inflammation. Yeah. All right. So how do we recognize pathogens or things that are meant to be in our body? Well, there's this thing called PAMPs on pathogens, pathogen-associated molecular patterns, and they are structures which are unique on the pathogen. And the most important thing is that that structure is important for survival of pathogen. So it can't just evolve away, right? So for example, flagella, LPS, double-stranded RNA. The reason why this is important is because bacteria and other pathogens in general evolve really quickly. And if we are trying to, as in our body cells, are trying to, recognize these pathogens or things that could possibly just, go away, it won't be a very effective way. So we want to kind of always make sure that we are trying to recognize structures that will always be present because they are essential for the pathogen to survive. And then there's DAMPs, damage-associated molecular patterns, and they are basically danger molecules released by damaged or dying cells, basically our body cells. So when they are, being infected or they're about to die, they will release these danger molecules, which will allow us to tell that there's pathogens around. And then there's the PRRS, or the pattern recognition receptors. And they are basically intra or extracellular structures on our cells, so our own body cells, that bind to the PAMPs and DAMPs. For example, there's toll-like receptors, which are a family of PRRS. So if you kind of see on the image here, it shows a good diagram. Again, you might not understand exactly what all of these means, but it's okay in the first time. But later, as you go on, you'll see that this diagram makes more sense. Now, inflammation is basically inflammation can be triggered by tissue damage, and inflammasome is triggered by DAMPs. So as we talked about, DAMPs is the damage-associated molecular patterns released by damaged or dying cells. And it basically produces active caspase 1, which cleaves pro-interleukin-1 beta into interleukin-1 beta, so the pro is removed, meaning it's active, which is a cytokine protein that promotes inflammation. So on the right side, there's a diagram, and just understanding this is more than enough for inflammation. All right, so now talking more about the second line non-specific internal defenses. So we have a cellular component. So these have PPRS. So phagocytic cells we talked about, inflammatory cells, and cytotoxic cells, all the cells we talked about. But then there's also other aspects such as soluble proteins. So cytokines, so interleukins, interferons, which help with signaling between cells. And then there's the complements. That's right. Yeah. Okay, yeah. Proteins circulating in blood. And these basically allow for direct killing or recruitment of innate cells to kill the cell or the pathogen. And then thirdly, there's inflammation, which we kind of talked about just before. And that just basically results in pain, heat, redness, swelling, and loss of function. Yeah, so complement pathway. So this involves the complement proteins that we were just talking about. And these are formed in the liver. And the mechanism of action, that's what MOA stands for, after recognition of pathogen surface structures. So after the complement recognizes the pathogen, which it can, it will either do one of these three things. One, coat the pathogen with the complement molecules, so it's easier for phagocytosis because it allows the immune cells to see that this cell is infected because there's complement molecules around it. Secondly, it can also enhance local inflammation through the release of complementary proteins, and this will attract more immune cells, hence get a better outcome. Or third, you can have direct killing of the pathogen with membrane attack complex, or called MAC themselves. So here is some ways. So there's also a name for these: classical pathway, alternative pathway, lectin pathway. You can memorize it in that, but I think in the end you will have to understand what they do. So basically, they could be cause inflammation, opsonization, and phagocytosis is basically coding them. Inflammation again, and lysis of microbe via MAC.
Okay, can I just add something in there?
Yep, sure.
So if you just go back one. So the way, an easy way I remembered it is all the A's cause inflammation. So C3A, C5A inflammation. And then I just thought, so the membrane attack complex is when C5b has a lot of other complements like 6, 7, 8, 9. They all form together and they put a hole in the bacteria. So that was quite like advanced. So it was higher up. So that was C5 and it had to be B because all the A's cause inflammation. Whereas C3b is just opsonization for the phagocytosis. Yeah.
That's a really good way. I should have used that when I was studying. But yep, there you go. All right, so now we're going to talk about phagocytosis. So we talked about this, we've talked about phagocytes, which are cells that engulf and digest pathogens. And that is basically the act of phagocytosis. So what happens is we detect non-self antigens and complement proteins on pathogens. And then phagocytes engulf this via endocytosis and digest the pathogen. I'm hoping you guys know what endocytosis means, but it's basically when we just get something extracellular and bring it inside. So the actual steps are basically the phagocyte binds to the target pathogen with antigen and complement protein receptors. So it recognizes that it's a non-self antigen, and then it binds to it. Then the pathogen is engulfed via endocytosis. So if you see here, the pathogen is recognized, and then it starts to get engulfed. Then it comes in. Then a vacuole forms and encloses the pathogen, and this is what we call a phagosome now. And then there's always there's going to be a lysosome within the phagocytic cell already, and this will fuse with the vacuole, creating a phagolysosome. And because these lysosomes are deadly and toxic, they will help ingest and digest the pathogen and release the waste products via exocytosis. So basically killing the cell and releasing the debris. And after that, the phagocyte will go on to engulf more pathogens. Now, and we'll talk about a bit more about what happens after for other cells in the adaptive immunity. But that's phagocytosis. So yeah, now there's this thing called leukocyte migration, which is basically immune cells called leukocytes or white blood cells. Because they need to move to infected tissues and lymph system for function, they have this kind of mechanism which allows them to do that. So firstly, there has to be initial exposure. So PAMS or damage recognition damage recognition by our body. Then macrophages will increase expression of adhesion molecules. And what these would mean, they will release cytokines and chemokines. These adhesion molecules will help of the immune cells basically adhering to the entrances towards the tissue, so allowing it to get in, basically. Endothelial cells will activate, and what this means is they will increase expression of surface adhesion molecules called selectin and other things, but selectin is the most important one. And this will promote the rolling of neutrophils. So I think I have an image here. So it's basically happening here, and integrin or selectin here. And basically it's like rolling in towards the tissues, basically. So rolling of neutrophils. Also, the circulating leukocytes respond to the endothelial cells being activated. And this leads to expression of selectin ligands, integrins, and chemoreceptors themselves. And this allows leukocytes to follow the chemokine gradient to affected tissue, meaning they get in towards the affected tissue. Then there's also the migration of the leukocyte itself. So the cells will bind to ligands and tissues, And histamine, which is going to also be released, will increase the permeability of the endothelium, which makes it easier for the leukocytes to come in to the cell, to the tissue. And at the site of infection, activated leukocytes respond to chemokine on endothelial surface. And this is done via the chemokine receptor, and it moves through the endothelium into tissue. And we call this the extravasation. So this is a really good diagram of it. Yeah, so basically we need this to happen so we can actually get our immune cells to the site that we need it for it to activate.
Can I add one thing there?
Yeah, sure.
So I don't know why this confused me so much, but last year, all you have to remember is integrin is on the leukocyte, whereas selectin is on the endothelial, the endothelium. So they will bind to each other. So the selectin makes it sticky, and that will so the leukocyte can attach to it, and then it can extravasate into the lumen. Yeah,
so like the cell, the leukocyte will have a selectin ligand, which will basically bind to selectin. Okay, so that is all of you need to really know about innate immunity. Now we want to go into adaptive immune response. Only half of it, because the other half we covered in the third lecture. So dendritic cells, these are the bridge between innate and adaptive. And these, basically, their role or function is to process and present pathogen/antigen on the MSC markers. And these are also called professional antigen presenting cells, or the main antigen presenting cells. And if you find them in epithelium, they are called Langerhans cells. That's just a different name if they are in the epithelium. So dendritic cells are very similar in mechanism to macrophages or any other phagocytes. So basically, dendritic cells will detect the PAMPs and phagocytose the pathogen. So it breaks down via the phagocytosis steps we talked about, and this will form debris or and which will also include peptides. And these peptides are shown on the MSC class 2 markers on the dendritic cell surface. And the dendritic cells will migrate from the site of infection, or where they first found the pathogen, to the lymph nodes where immature B and T cells are located. And at this lymph node, it will find and activate specific CD4/T helper cells, and hence activate the adaptive immune response. So this is why it's called the bridge between innate and adaptive, because it starts off being innate, like acts like a macrophage, detects the PAMs and just phagocytoses it for every pathogen. It's not like it will be different to a specific pathogen, but then it will present a peptide, so a degraded protein basically of one of the pathogens, and present it on its MSC class 2 markers, which we'll talk about a bit later. And then they will migrate to a lymph node where there's immature B and T cells, and activate specific CD4 T helper cells, which basically activates the adaptive immune response. This is a bit of a diagram here. So basically, this is a DC or dendritic cell. Phagocytosis degrades protein peptide presented on its MSC class 2, migrates all the way to a lymph node where there's T and B cells, and activates the adaptive immune response. So what is a MSC marker? MSC stands for Major Histocompatibility Complex, and it's basically made up of proteins or antigens. And there's two types. Type 1 is MSC class 1, and these are present on all nucleated cells. And these interact with CD8 plus or cytotoxic cells and natural killer cells. A way I think it was PSB Pingu last year had to memorize with which class acts with CD8 or which class acts with CD4 is that the rule of 8, I think. So to get to 8 from 1, you have to times 8. That's why class 1 interacts with CD8, whereas class 2, to get from 2 to 8, you need to times 4. That's why class 2 acts with CD4 plus. That's a way that could help you memorize it. It's up to you, but I found that pretty useful. So the MSC class 1 pathway has the pathogen source from inside the dendritic cell. So what that means is if a virus infects a cell, this MSC class 1 pathway will kill the infected cell. Whereas MSC class 2 markers are present only on antigen-presenting cells, which will include dendritic cells, macrophages, B and T cells. But mainly we want kind of more interested in DC. These will communicate with T cell antigen receptors, TCR for short, on CD4 plus cells to be activated, as we talked about. The MSC class 2 pathway, for these, the pathogens will be sourced from outside the DC. So it's like extracellular pathogen is eaten by the APC. And then this APC will activate the immune response. Whereas for class 1 pathway, a virus infected cell will be killed itself. And CD8 cytotoxic cells, they go out and kill infected cells. That's what their job is. And they only kill ones that present the same MSC and peptide signature antigen as the one that they were shown to. Whereas CD4 or T helper cells, they help other cells like B cells to create the best antibody or being specific to the antigen they were showing. So this is a pretty good diagram right here showing how it works. So there is a T cell and they will differentiate. There's CD8, we talked about CD4, and they will be activated by the APC. So class 1, actually, so not APC, so it will be activated depending on the cell. So for CD8, it will be activated by any antigen presented on MSC class 1. And MSC class 1 is present on all nucleated cells. So it could be anything, to be honest, technically. Whereas for CD4 cells, it has to be antigen presented on MSC class 2, meaning it will be an APC. And then CD4 becomes T helper cells, where CD8 becomes cytotoxic cells and destroys infected cells right away. And T helper cells will go and activate B cells to proliferate, but also helps with activation of T cells as well. And B cells will secrete antibodies. I think I'll go into a bit more detail here. So cell-mediated immunity is the type of immunity which involves no antibodies. Again, the med course, the lectures you will get, I don't think they mention cell-mediated immunity and humoral immunity at all. They just talk about T cell activation and B cell activation. But I found it kind of useful still having these categories because it is something that exists. It's not like this is like non-existent. But yeah. So basically, cell-mediated immunity, there is no antibodies. And it mainly involves the T lymphocytes or T cells. And basically, what happens is that dendritic cells phagocytose pathogens and present the pathogens and antigens on MSC class 2, as we talked about, and intracellular molecules on MSC 1. So they present antigens on both MSC 1 and 2. MSC 1 marker is again presented to CD8 plus or cytotoxic cells, whereas MSC 2 markers are presented to CD4 plus or helper cells. Then the activated CD4 plus cell produces cytokines, which will allow for expansion and differentiation of activated CD8 cells. And CD8 plus cells will find cells presenting the same class 1 MSC and peptide signature as itself. Why? Because the dendritic cells phagocytose the pathogen and they are trying to find the same one that it basically presented them to. One thing that is important to note is that all T cells require 2 signals to become fully activated. So a TCR/MSC peptide and co-stimulation. So this will help prevent non-specific activation, which will be very dangerous if that happens. For humoral immunity, this is immunity involving antibodies. So there is antibodies. This will involve B cells, but also T cells. So how it works is that dendritic cells will undergo phagocytosis and present the peptide on MHC class 2, again, as previously. Then the dendritic cell will find a specific CD4 plus cell or helper cell, activating it. Then the right B cell with a complementary receptor, BCR, basically means the B cell receptor, to the CD4 plus, so the specific CD4 plus that was activated. This will activate by the help of the helper T cells. Then the B cells will undergo clonal expansion and multiplies, becoming plasma B cells, which will then produce and secrete antibodies. So clonal expansion is basically when there is a production of plasma B cells, which basically produce and secrete antibodies to specific to the antigen that activated it, but also it produces B memory cells, which allows for faster, stronger, and longer immune response when a reinfection occurs with the same antigen. And this is basically the basis of vaccination, right? Because if you get vaccinated, you're exposed to it once, and when you get exposed to it once again, you have a faster, stronger, and longer immune response because of the memory cell. Okay, so for the antigen receptors themselves, there's TMB antigen receptors, and these are created by each of the corresponding cells. So T cell receptors will be created by T cells, B cell will be created by B cell receptors, or B cell receptors will be created by B cells. They have a defined set of genes. So yeah, and the cells, or can't see, but basically there's a variable region, and these are encoded by V for variable, D diversity, and J joining segments of the genome. And there's also a constant region which is encoded by the C segment. So this is talking about heavy chains, and light chains is talking about antibodies. So heavy chains will have all of them, so V, D, and J. Light chains will only have V and J segments. Going back to the T cell and B cell receptors, T cell receptors only bind to the MSC plus peptide combination, so they require APC. Whereas B cell receptors, these are receptors secreted when B cell is activated, right? The plasma B cells, aka antibody or immunoglobulin, that's also what it's called. Hence, receptors can bind to things as they appear. They don't need APCs. And antibodies work in very similar fashion to complement proteins that we talked about before. They can optimize, they can flag for phagocytosis and things like that. Interestingly, B and T cells also interact with each other via their TCR, that should be T cell, sorry, and MSC/peptide, which will be the B cell. So the B cell in this case will capture the antigen using its surface BCR. Then the B cell will present the antigen to the T cell. Okay, so they can also interact with each other. Antibodies. So these are different types of antibodies. I don't think you need to know too much of it. It's not that hard to memorize some of the different characteristics, but don't need to know this, I think. You might just need to roughly read through these and see where you come across. You just learn kind of as you go. Like, depending on the disease, you'll realize that IgA is the more common one, IgE is the one that's affecting, things like that. But just know for now that there is these different types with different mechanisms. So basically, this is a generic immune response. So there's 5 steps to it, basically. Pathogen is captured, and DC migration happens to lymph nodes. And basically, this is when the pathogen or antigen is exposed to the B or T cells and presented, and this will lead to T cell activation and also B cell activation, and then this will result in T and B cell effector functions. So the effector cells in this case will be the cytotoxic cells for T cells and the B plasma cells for B cells. Yep, so this is a really, really simple diagram. You will need to know more than just this, but this is a really good idea in terms of big picture. And that's all. Any questions? Sorry, I wasn't. Oh, if there's no questions, we can move on to physiology.
Thanks so much, Peter. If it's okay, would you mind if I share screen for physiology? Just so I can do some writing.
Yeah.
Cheers. Great. Can you guys see all of that? Okay, so physiology is a bit terrifying at first. Like David said earlier, you're gonna find a lot of it confusing, especially because they sort of just throw into neurophysiology without any context. So hopefully we can help you guys make some sense of it. First of all, just by briefly going over what the nervous system actually looks like. So yeah, let's start off with that. So these are some key terminology that you need to be familiar with. This isn't that important for this week, but when you do go spinal cord stuff later on, you can sort of look over that. Yeah, so look. It just crashed. Let's see. There we go. Yeah, so let's start off with the different divisions of the nervous system. So we have the central nervous system and also the peripheral nervous system. So your central nervous system is your brain and spinal cord, and that is sort of where all the information processing happens. And we also have our peripheral nervous system, and this sort of communicates with our central nervous system. So your peripheral nerves are everything outside of your spinal cord and your brain. And with our peripheral nervous system, we can separate into the sensory division and motor division. Sensory division, that's also called afferent. It's basically where we're sending information to our brain. And our motor or efferent division is where we're sending information from our brain to effector organs. So for example, sending commands to our muscles so we can execute movements, and also to our glands as well. And also our motor efferent division can be further split into the ANS and the SNS. So think of your ANS as sort of like your digestion or your breathing, heart rate. It's involuntary. You don't really think about it. You can't control it. And think about your somatic nervous system as sort of your arms and limbs. So you can voluntarily control movement. Finally, our autonomic nervous system, we can split it into the sympathetic division and also the parasympathetic division. So sympathetic division, that's our fight or flight response, and parasympathetic division, rest and digest. So they're sort of like two opposite sides. A brief overview of the difference between sympathetic and parasympathetic. So remember, this is part of the autonomic nervous system. So these are things that we can't control voluntarily. So sympathetic, fight or flight. So what that does is it dilates our airways, dilates our pupils. So you think about if you're running from danger or about to fight somebody, you're going to want to have better vision, better respiration, and your blood flow is also going to increase to your muscles and decrease to your skin. Heart rate's going to increase, and your gastrointestinal tract is going to have inhibition, because you don't want to be sort of like digesting foods when you're in a fight or flight response. And on the opposite side of the coin, we have parasympathetic, and this is our rest and digest. So we have bronchoconstriction, pupil constriction, vasoconstriction, and our heart rate is decreased to conserve energy. And this is where more digestion happens, because we have time to sort of like send our blood flow to help digest food. So I'll briefly go over this. This isn't too important at the moment. So cells of the nervous system have two main types of cells. We have nerve cells and glial cells. For today, we'll mostly be focusing on the physiology of nerve cells. So you can have a read over these in your own time. So nerve cells can be classified either by structure or function. So structure, we have unipolar, bipolar, multipolar, pseudo-unipolar neurons. This isn't that high yield, and once again, you can just go over it in your own time. More important is the function. We can classify neurons by three functions. So sensory neuron conveys sensory information from receptors to spinal cord and the brain. We have motor neurons, which conducts impulses from the brain by the spinal cord to effector organs. We also have interneurons, which connect sensory and motor neurons.
Jono, so just, you might want to share screen again, because I think for a few of us, there might be like black rectangles stuck on the screen for some reason.
Oh, right, okay. Cheers. Is it better now?
There's still one in the bottom left corner for some reason, but the other one's gone. There were two before.
Right, is it still there now?
Yeah, I'm not sure why it's there.
Can you see our faces on it?
Yeah, I've moved that one.
Okay, that should be fine, I think. If you try minimizing our faces, and if that does, yeah, there it's gone.
Yeah, so on the left, we have the different structures, and on the right, we have neurons classified into different functions. So we have a sensory neuron, which has receptor endings. So it could be like touch receptors, and it sends it to an interneuron, which connects the two. And we also have a motor neuron, which sends information back and can sort of convey signals to effector organs such as muscles. And the other type of cell is the glial cell. That's not too important at the moment, so you can have a read in your own time. Okay, so moving on to neurophysiology now. So first topic is resting membrane potential. So basically, we're looking at what is happening inside a nerve cell, basically. So first of all, we have different types of membrane proteins. So importantly, with ions, as they are charged, they're not able to diffuse through the plasma membrane. And so that's why we need membrane proteins to facilitate their diffusion. So we have passive transport. This doesn't involve energy, and it involves molecules diffusing down their concentration gradient. So we have just normal diffusion, which is through the plasma membrane, but ions need channel proteins, or also called facilitated diffusion. So some examples of these are voltage-gated channel proteins. So these open and close with changes in voltage. We have ligand-gated. So these open and close with the bonding of ligands. So for example, neurotransmitters or hormones. We also have mechanically gated channel proteins, and these open and close due to physical pressure, temperature changes. And we also have leaky channels. And so these are just basically channels that are always open. We also have active transport, and this involves using energy in the form of ATP. And this also helps to pump molecules against their concentration gradient. So the key thing here is pump proteins. So an example is the sodium-potassium pump. So what the sodium-potassium pump does, it swaps 3 sodium ions for 2 potassium ions. So it pumps out 3 sodium into the extracellular space, and it pumps in 2 potassium into the inner cell, and it uses up ATP in the process. So over here, you can see we have our active transport, which is using ATP. And then we have our 2 types of passive transport, diffusion and also facilitated diffusion. Okay, so moving on to resting membrane potential. So what is membrane potential? So membrane potential basically compares the charge inside the cell to outside the cell, okay? So if you imagine we had 2 electrodes, right? We stuck one outside the cell and we stuck one through the plasma membrane inside the cell. And basically, if we measure the charge difference across the membrane, that is our membrane potential. Now for a nerve cell, so this is sort of a relative measure between inside and out. So if we take the extracellular fluid to have a voltage of 0, so it turns out that in the intracellular space, cells actually have a negative charge. And if you were to stick 2 electrodes across the plasma membrane, you get this measure of 70 millivolts. So basically there's a potential difference across the membrane of 70 millivolts. And because it's more negative on the inside, and because we measure relative to the outside, so we do 0 minus 70 and we get negative 70 millivolts as our resting membrane potential. So why is there a difference in charge inside the cell compared to outside the cell? Well, it's basically due to differences in the concentration of anions and cations across the cell membrane. So quickly about polarization, right? So if the potential difference across the cell membrane is non-zero, so if there is a potential difference, then we say the nerve cell is polarized in this case. So if our membrane potential becomes more positive than our resting membrane potential, remember we said that was around negative 70, so it's getting closer to 0, then it's getting depolarized. But if our membrane potential gets more negative than our resting membrane potential, so over here in hyperpolarization, we're going to call it hyperpolarized because it's becoming even more polarized. So we have resting potential. Depolarized is where we get closer to 0 or more positive, and hyperpolarization is where we get more negative. So what is actually responsible for this membrane potential across a nerve cell? So it's mainly due to differences in concentration of ions, like we said. And what causes these differences are sort of 4 different factors. First of all, we have the sodium-potassium pump. We have ion channels. We have what we call an electrochemical gradient. So we have 2 forces at play when looking at the physiology of a nerve cell. So we have electrical. So obviously, this is where a positive and a negative ion would attract, whereas a positive and a positive would repel. Similarly, a negative and a negative ion would repel. But we also have chemical forces at play. So as we know, molecules will want to diffuse down their concentration gradient. And so these 2 forces, we call them together the electrochemical gradient. Finally, the selective permeability of membranes is also responsible for creating a membrane potential. So let's first look at some of the ion concentrations for a nerve cell. So if you sort of do a cross-section of a nerve cell, measure the concentrations, you'll find that the concentration of sodium is small intracellular and high extracellular. The concentration of potassium is high intracellular and low extracellular. So sodium is high inside this, high outside the cell, and potassium is high inside the cell. So they're sort of opposites. We also have chloride, which is high extracellular, and organic anions. So basically, these are your proteins that are produced within the cell. And these are usually negatively charged, and we have a high intracellular concentration. And these proteins, they don't exist outside of the cell. So let's first look at what if we just had a cell with only potassium and basically no other ions? What would happen? Well, basically, our active transport, so our sodium-potassium pump, it's going to pump in potassium. Remember what you said, pumps in 2 potassium for every 3 sodium that is pumped out. And this is going to create a high potassium concentration inside the cell. And there are also ion channels, so our leaky channels for potassium that are present. So what this means is that our potassium is going to diffuse down its concentration gradient through the membrane until it balances the electrical force. So I've got a few diagrams on the next page which will illustrate this better. And finally, selective permeability of the membrane is going to prevent the anions from also moving with our potassium. So the ions will move down the electrochemical gradient until we reach equilibrium potential. And equilibrium potential is where those two forces, electrical and diffusional, are basically equal and opposite. So I've got a few diagrams here. So in the first diagram, this is our starting state. So due to our active transport, we're going to have more potassium ions inside the cell than outside of the cell. But each sort of potassium is balanced with an anion, and so our membrane potential is going to be zero. So there's zero net charge inside the cell and zero net charge outside of the cell. But potassium is more concentrated inside the cell and less concentrated outside of the cell. We also have these channel proteins here, these leaky channels, and they're only selectively permeable to potassium. So it means that our anions aren't going to be able to flow through, but our potassium is. So if we move over to this diagram, right, well, we have our two forces. So our chemical force, which is our diffusion gradient, it's going to be driving potassium outside of the cell. And so as potassium moves outside of the cell, there's a charge imbalance. So we're going to get more positive charges outside of the cell and less positive inside the cell. So our membrane potential is going to become more negative. And our anions basically can't move out. Also remember, we have a selectively permeable membrane, and it's only these channels which are allowing the potassium to move through. Then finally, as more and more potassium ions flow through, while the right-hand side, which is outside of the cell, is going to become more positive, and the inside of the cell is going to become more negative. So even though we have that diffusion or chemical force, we now have an electrical force that is acting in the opposite direction. So if the outside of the cell now becomes more positive, then it's going to repel the sodium that is sort of trying to flow through in that case. So this is going to become more positive, and this is going to become more negative. So we have a chemical force that is driving it in this direction, but an electrical force that is counteracting that and driving potassium ions in the other direction. And so when these two forces balance each other out, we're at equilibrium. And the membrane potential, when we are at this equilibrium, is what we call the equilibrium potential. So as you can see, due to a selectively permeable membrane and also the differences in concentration, we have a charge difference across the membrane. And so what actually affects this equilibrium potential? Well, it's the difference in concentration inside and outside of the cell. So for example, if we look at potassium, if it's 5 millimoles outside of the cell and 140 millimoles inside the cell, which is normally what it is, we have an equilibrium potential of negative 84 millivolts. Similarly with sodium, because the concentration difference is a bit less, it's going to be positive 66 millivolts. So looking at the resting membrane potential now. So the difference between the resting membrane potential and the, let's clear the drawings, there we go. So the difference between the resting membrane potential and the equilibrium potential is that the resting membrane potential factors in all ions. In reality, we have more than just potassium in a cell. So the RMP is going to be our weighted average of the equilibrium potential of all the ions. And the weight that we give to each equilibrium potential also depends on the membrane permeability. So during your lectures, you might be given this very terrifying equation. The good thing is that we don't really get tested on it. And it really is just sort of a mathematical representation of what we said here. So don't worry too much about that. So to summarize, let's sort of go from the start and look at what happens and what creates this resting membrane potential. So let's imagine we start off with a cell where the concentration of ions are equal inside and outside the cell. So the cell is going to be producing organic anions. So these can't diffuse. So it's going to become more negative inside. So our resting membrane potential is going to become a bit negative. So for example, maybe -5 millivolts. So the sodium potassium pump, that's going to be pumping out sodium and pumping in potassium. So we're going to have a high extracellular sodium concentration and a high intracellular potassium concentration. And so there's differences in permeability between potassium and sodium. So potassium is about 25 times more permeable than sodium. So potassium is going to leak out faster through the leakage channels down its electrochemical gradient. And so as the positive potassium ions leak out, our resting membrane potentials become more negative. At the same time though, sodium is going to leak in down its own electrochemical gradient. But because sodium is less permeable, this is going to be a slower process. So the RMP is going to become less negative, although not as much as the effect from potassium. So it might stabilize around -70 millivolts. And so this is basically how we reach our RMP of -70 millivolts. It's a bit of a simplification since we've ignored other ions such as chloride and calcium, but that's sort of like the big picture. Yeah, we'll just skip that. And moving on to action potentials now and how an action potential propagates. So the main way that cells actually communicate, nerve cells communicate, is through action potentials. So there are two important channels involved here: your voltage-gated sodium channels and your voltage-gated potassium channels. So voltage-gated meaning that they're opened or closed depending on the voltage. We have 3 states for our sodium channel: closed, open, and inactivated. Whereas we only have 2 states for our potassium channel, which is closed or open. And our sodium channel, it opens and closes rapidly, whereas our potassium channel, it does it slowly. And this is sort of just a review of polarization, depolarization, hyperpolarization. And we're also going to be introduced to repolarization. This is when the membrane returns to our RMP after we depolarize. Okay, so first of all, we have something that is called the threshold potential. So this is the point at which a self-sustaining depolarization takes place. Okay, so around -50 to -55 millivolts. In other words, this is basically where our voltage-gated sodium channels are going to open. So that's the threshold potential. And that flows on to our actual mechanism of the action potential. So we start off with our neuron, which is at our resting membrane potential. Remember, we said this was around -70 millivolts, and we looked at how that RMP was created previously. Both channels are going to be closed, our sodium and potassium channels. And we're going to have this threshold potential. So basically, once we reach -55 millivolts, our sodium channels are going to open rapidly. And this is going to result in depolarization. So remember, we have a high extracellular sodium concentration and a high intracellular potassium concentration. So what is going to happen when these sodium channels open? Well, we're going to have a massive influx of sodium ions because we have a high concentration outside of the cell. And so it's going to want to diffuse down its concentration gradient. So as positively charged sodium ions move inside of the cell, well, we're going to get depolarization as the inside of the cell becomes more negative. So looking at this diagram on the right, this upward spike over here, as our membrane potential depolarizes, basically becoming less negative and positive, that is when our sodium is flowing into our cell. So we said that our sodium channels close and open rapidly. And so as our membrane potential becomes more positive, this triggers our sodium channels to close, and we reach our maximum depolarization. At the same time, this positive voltage then triggers our potassium channels to open. So this happens more slowly. And remember, with our potassium channel, we have a high intracellular potassium concentration. So when the channel opens, our potassium is going to flow out of our nerve cell. And so as our positive ions flow out, there's a charge imbalance again. And so the inside of the cells can become more negative. And so that represents this dip over here once again. So this dip, where we are repolarizing, we're returning to our RMP, represents potassium channels opening and potassium flowing out. And because these potassium channels, they close and open slowly, right? They remain open for a longer period of time compared to sodium. So this is going to result in hyperpolarization, or what we call undershoot. This basically occurs because our potassium channel can't close quickly enough and too much potassium escapes. And then finally, our sodium potassium pump, it's going to help our neuron to return to our resting membrane potential of -70. And it's going to pump back out the sodium that flowed in and pump back in the potassium that flowed out. So during this time, both our sodium and potassium channels are going to be closed. So this is the action potential. So another diagram to visualize what is happening with the action potential. So at our resting potential, we have our sodium potassium pump maintaining that. During depolarization, we have our voltage gated sodium channel opening, and this is going to result in an influx of sodium. During repolarization, we have our voltage gated potassium channels opening. This is going to result in an outflux of potassium. Finally, when both closed, our sodium potassium pump is going to help us return to our resting membrane potential. And this is just another diagram to show in which direction our ions are moving during an action potential. So with our action potential, we have something called a refractory period. So this is basically where we cannot send another signal. So an absolute refractory period occurs because of our sodium channels being inactivated and unable to refire. And so this is represented by this black bar over here. So when an action potential occurs, we can't follow it immediately by another action potential because our sodium channels have become inactivated. Relative refractory period follows. This is where it's hard to start another action potential, but not impossible. And it becomes more difficult to fire a second action potential because, remember, our nerve cell is going to be hyperpolarized, or basically that undershoot. So it means that we're going to require more stimulus to reach our threshold potential of around -50 to -55 millivolts. So what does this mean? So once again, we have our potassium voltage channels closing too slowly, which results in hyperpolarization. And so it means that our action potentials can only travel in one direction due to the refractory period. So if our sodium channels are in an inactive state, then an action potential can't sort of fire in that direction again, whilst our potassium channels are still open. Finally, we also have two different electrical signal types. So we looked at action potentials, but we also have greater potentials. And so graded potentials, they're local, smaller charges, changes in membrane potential, whereas action potentials, they're rapid, large changes. You can think about graded potentials as sort of like small, local, short distance signals, whereas action potentials, they're all or nothing. Graded potentials spread bidirectionally, whereas action potentials only propagate in one direction, and that's due to that refractory period. And graded potentials are actually how action potentials can be initiated, and action potentials propagate throughout the entire membrane. So this is a diagram of a graded potential. So graded potential, it's a local area of depolarization. So what causes these channels to open? Well, it could be neurotransmitters, and it's going to allow ions to flow in basically. And it's going to allow sort of depolarization to occur bidirectionally. So to create an action potential or reach our threshold of -55 millivolts to fire an action potential, we actually need multiple graded potentials. And so a process called summation is required to hit this threshold. So we have basically excitatory presynaptic neurons and also inhibitory presynaptic neurons. So if you think about the neuron that's sort of previous to our postsynaptic neuron, then it's going to be firing multiple sort of action potentials, and it's going to be releasing its own signals. So there's two ways that we can generate enough depolarization to reach our threshold. So one is spatial. This is where we sort of have two neurons sending an impulse at the same time, and it's going to add up together to reach our threshold. We also have temporal. This is where a single neuron sends sort of successive impulses, and this is going to also add up to reach our threshold. And with our excitatory and inhibitory neurons, if they both fire a signal at the same time, these can cancel out, and it won't result in any depolarization. Yeah, so sorry, that was very long and may have been confusing. Was there any questions from anyone in the chat? No. Okay, I think I'll pass it on to Andrew then.
Okay. I'll just share screen. Just 1 second. All good? Yeah, okay. So now we'll be talking about synaptic transmission. So first of all, what is a synapse? So a synapse is basically where one neuron kind of like touches another neuron or another cell. So it's a junction between a neuron where it contacts and communicates with either another neuron or another cell. So that might be a muscle cell or maybe a glandular cell. And so you can also describe the components as presynaptic and postsynaptic. So because you have one cell touching another cell. So one neuron touching the second neuron. The first neuron is presynaptic, so it's before the synapse. And the second neuron or the cell will be postsynaptic because it's after the synapse. So you can also have two types of synapses. So they can either be electrical or chemical. So basically, if it's electrical, the signal that the first neuron sends to the second neuron will be electrical. If it's chemical, they'll be basically releasing something called neurotransmitters or chemicals and communicate chemically instead. So if you look at the bottom on the left-hand image, so what happens is you have your action potential from the neuron. So what Jono just talked about, the action potentials, they can arrive to the presynaptic terminal or the axon terminal. And what happens is they'll activate something called voltage-gated calcium channels. So voltage-gated meaning they'll only activate when a certain voltage is reached. So that will be activated by the actual potential. So the calcium channels will open, and then that will let calcium ions into the axon terminal. What happens is the calcium can then trigger synaptic vesicles. So vesicles are basically little storage thingies. So they're like the circles that you see in the terminal, and they contain the bunch of neurotransmitters. So what happens is your calcium will trigger your synaptic vesicles, and then your synaptic vesicles will basically they'll basically release their neurotransmitters. They'll kind of merge with the cell membrane and basically vomit out all the little blue dots, which are your neurotransmitters, as you can see in that image. And then these neurotransmitters will sort of diffuse across the gap, and they'll bind to receptors on the other cell. And yeah, then that can cause another action potential to be generated if it's another neuron, or it might cause the muscle to, you know, contract. If it's an electrical synapse, rather than having chemical signals, it will essentially just be electrical currents flowing through these two kind of channels you see. So these channels are called gap junctions. So just another diagram for you here. As you can see on the left-hand side this time, we have what are the electrical synapses. So this is the gap junction. And you'll basically have ions flowing through, and that will be your electrical current. And then on the right-hand side, you'll see what again are your chemical synapses. So as you can see, the vesicles have fused with the presynaptic membrane, and it's vomited out all its neurotransmitters, and they're diffusing across to bind with the receptors, and this will let in like ions and stuff to activate another action potential. So something to note is that electrical synapses and chemical synapses will have fundamental differences. So first of all, obviously, with electrical synapses, you have current flowing through gap junctions. So these gap junctions connect the two neurons, and it basically lets the current flow through the two neurons. And you have chemical synapses, which communicate through the neurotransmitters, as we've just talked about. And as you can imagine, electricity will be comparatively fast. So you have electric current, and because the gap junctions connect the two neurons, there's a very little distance to travel. So in addition to the current being faster, there's also less distance. Comparatively, the chemical synapses are slower. So this is largely because in chemical synapses, you just rely on diffusion, and there's also a larger distance to diffuse. So if you have a look at, oh, that shouldn't say gap junctions, that should just say gap, sorry, in the third row. The gap is about 1 to 3 nanometers in electrical synapses, and the gap is about 20 to 50 nanometers and up to 1 millimeter in chemical synapses. And electrical synapses, the current will simply just transmit information. So the current won't get stronger, and it will get a little bit weaker as it travels with distance, but it's basically the signal that it transmits, it won't get stronger. But if you have chemical synapses, when the chemicals diffuse across, they will amplify the signal again. So that's just something to note. And another thing to note is that electrical synapses, the current can travel both ways. So it could travel from the first neuron to the second neuron, but it also could travel from the second neuron to the first neuron. However, with chemical synapses, only the neurotransmitters will only be basically sent one way. It won't be sent the other way. So it will always be the first neuron sending neurotransmitters to the second neuron. It won't happen the other way. So normally you find electrical synapses between neurons. So it will be neuron to neuron. But with chemical synapses, you can have them neuron to neuron, or they can go to a cell, for example, a muscle cell. So the last row, that's just basically saying your neurons and your electrical synapses, they're connected by gap junctions. The synaptic cleft, which is like the little space you see in the chemical synapse, that will be filled with basically extracellular proteins that, you know, just basically provide structure. Not a very important point, but just something to know. And this is basically something we've already gone through. It's just how a chemical synapse works. So we'll go through it just relatively quickly again, because it's quite important to know. So what happens is at the presynaptic terminal, your action potential arrives. This will trigger your calcium channels and let calcium into the axon terminal. and that would just be through diffusion. And then the calcium will act as an intracellular messenger. This will bind with proteins, and this will cause the synaptic vesicles to fuse with the axon membrane. So calcium is then removed from the terminal, because if it remains, it'll keep attaching to the proteins, it'll keep causing neurotransmitters to be emitted, and you don't want that. So you get rid of the calcium once it's done its job, it's taken up by mitochondria or via an active pump. Neurotransmitters then diffuse across the synaptic cleft, and it binds to specific receptors. And then this will open ion channels and result in another action potential being generated in the next neuron or cell. And the neurotransmitters can be removed through 3 methods. So the first method is reuptake. So basically the presynaptic terminal or astrocytal glial cells, which are like supporting cells for the neuron. They're not as important, but basically they can reuptake the neurotransmitters and they can be recycled. Another thing that can happen is degradation. So you have enzymes that are on the postsynaptic membrane, and they will break down the neurotransmitters once they've done their job. And the third option is just simple diffusion. So they can just diffuse away from the synaptic cleft and they won't have any action anymore. This is just a diagram, outlines the whole process once again. So you can look through this in your own time if you'd like. And something to note is something called the synaptic delay. So this basically refers to what we talked about before, that chemical synapses are comparatively slower to electrical synapses. So chemical synapses about 0.3 to 5 milliseconds, whereas electrical synapses just take up to 1 millisecond. And yeah, you don't need to know these numbers, of course, but it just reflects the time needed for neurotransmitter release, the diffusion across the cleft, and to bind to receptors. And synaptic cleft is the rate limiting step of neurotransmission. So in other words, it's the slowest step in transmitting a signal. So the fact that chemical synapses are slower than electrical synapses will be emphasized a lot. A bit more on neurotransmitters. So what really are neurotransmitters? So they're basically substances released by the neuron for chemical communication. That's all they really are. They're just a bunch of substances for communication that the neurons will release. So there's a wide variety of neurotransmitters. There's different types. Some neurons will have multiple types. Some will only produce one type. That's just something to note. And beforehand, before they're released, neurotransmitters will be stored in the presynaptic terminal, in the vesicles that we mentioned, so the little circles that we saw before. And their release is calcium dependent, so they need calcium to be released. And these neurotransmitters, they can be either excitatory or inhibitory, depending on the postsynaptic cell. Afterwards, they need to be eliminated, as we talked about through the methods just before, reuptake, degradation, and diffusion. Because if they remain there, they will continually exert influence on the cell, and you don't want that. Something else to note is that they're synthesized in advance. So large peptides are made in the cell body and transported to the terminal. Small neurotransmitters are made in the cytosol of the presynaptic terminal itself. A very minor point, though. As we talked about, the neurotransmitters can be either inhibitory or excitatory. So if it's excitatory, that means it will produce a graded potential in the postsynaptic neuron. It will depolarize the neuron and bring it closer to threshold to produce the actual potential, the action potential, sorry. But if it's inhibitory, then it will hyperpolarize the postsynaptic neuron. And this will lower the likelihood of it reaching the threshold to produce an action potential. And then you can have different types of receptors on the postsynaptic neuron. So you can have what is called ionotropic. This basically means the receptor itself, the receptor for the neurotransmitter itself, is the ion channel that opens when activated. So generally, they will mediate rapid postsynaptic effects, but the effects are generally short-lived. Or you can have something that's called metabotropic receptors. These receptors are separate from the ion channel. So the activated receptor will affect the ion channel through intermediaries instead. These might be something called G proteins or intracellular messengers. So these typically amplify the response of the initial neurotransmitter. So they will amplify the signal, but these will give slower responses. And this is just a diagram showing them too. as you can see on the left-hand side, that's your ionotropic receptor. So what happens is your green little circles are neurotransmitters, they'll bind and then they'll open and let the ions flow through. But with your G protein coupled receptors, what happens is the receptor, it binds to neurotransmitter, and then once it's activated, it will tell its little G proteins to, go to the channel, to the ion channel. So the ion channel isn't part of it, it's separate. But once the G proteins go to the ion channel will open, and then your neurotransmitters can flow through. Not your neurotransmitters, but your ions can flow through. And now we'll move on to some like spinal cord stuff. Unfortunately, the lecture we had last year was quite messy. Hopefully yours is different. But yeah, hopefully it won't be too messy for you guys. But yeah, it was quite bad in our year. So we'll start off with reflexes. So your reflexes, they're involuntary and almost instantaneous response to a stimulus. And this response is generated at the level of the stimulus. So it doesn't travel up to your brain and you don't think about it. It's just a reflex. It's something you do automatically. And the reason why is it's to save time. For example, if you touch something really hot, you don't want to get burned. And the longer it takes, the more you'll get burned. So you want to minimize the time basically, and reflexes let you do that because it doesn't travel up to your brain and it stays at the level of the spinal cord. So a reflex arc will consist of 5 components. So the first is your sensory element. So that'll be your sensory receptor. That'll be your afferent nerve, your sensory nerve basically, and that will maybe sense the heat. For example, if you touch something really hot, that'll be your heat receptor. And then the afferent nerve itself is the second component. It will take up the information to your spinal cord and brain. Not your brain, sorry, just the spinal cord. And it will process this information. And then once it processes this information, it will give it to an efferent nerve, which basically takes signals from your spinal cord, from your CNS towards your muscle or your effector organ. And then finally, when it reaches the effector organ, like your muscle, it will tell it to pull your hand away from the burning pot or something like that. So that are the 5 basic components. Okay, that's gone way too many times. Okay, there we go. And you have what are called monosynaptic and polysynaptic reflexes. So monosynaptic just means there's one synapse in the reflex. So if you look on the left-hand side, you can see that the sensory neuron, it travels from your leg all the way to your spinal cord, and then it synapses to your motor neuron. And then your motor neuron travels back to your leg and, pulls off the reflex. It tells your leg to move or whatever. And you can see that there's only one synapse between the sensory neuron and the motor neuron. So that's monosynaptic. If it's polysynaptic, then you might have an interneuron in the middle. So on the right-hand side, you can see that your sensory neuron goes to your interneuron, and then your interneuron then synapses with your motor neuron. So polysynaptic means anything that's 2 or more synapses in the reflex. And what the interneuron can do is for a bit of extra processing, and it can also go up to the brain at the same time to relay some information. And then we'll have a basic look at what are the nerves in the spinal cord. So first of all, you have what's called gray matter. So that's the butterfly looking thing you can see in the middle of the spinal cord. That's a bit darker, so hence why it's called gray matter. So this contains cell bodies of neurons, and that includes your motor neurons and your sensory neurons. So the axons, which is like basically the long bits of the neurons, the axons of the neurons here are myelinated. So what myelin is, it's like basically a fatty layer on the neuron itself that helps with insulation, protects the neuron, nourishes it, etc. But here the axons in the gray matter part, they're unmyelinated. The white matter parts, however, they have more myelinated fibers, so they'll have that fatty sheath. And this helps with basically protecting it and all of that stuff. And so in the white matter, you'll find what are called spinal tracts, which we'll go through a bit more. But first of all, we'll talk a little bit about the dorsal root ganglion and all of that stuff. So what the dorsal root ganglion is, it's this kind of bulbous thing here. So this little bulbous thing will contain all of the sensory nerve bodies. So all of your sensory nerves that come from the periphery of your body, you can find the nerve bodies here. And the dorsal root here is basically where you'll find your incoming sensory fibers. So you might have like, I don't know, an arm here. That's not really an arm, but just pretend it is. You'll have sensory fibers, perhaps something touched you, and then the sensory fibers will come in. It'll come in through your dorsal ramus. You don't really need to know what a ramus is, it's just a name, but dorsal just means posterior, behind basically. So dorsal means behind, ventral means in front. But yeah, so it'll come into your dorsal root ganglion. The nerve body will be here, and then it'll continue on through the dorsal root and into your spinal cord. And from there, it might go to your brain or something. And then you also have your ventral root. So ventral root is here. And so your ventral root contains your motor fibers. So your motor fibers are what goes to your muscles, and they're what tells your muscles what to do. And so, you know, you might receive a signal from your brain, it comes down onto your motor neuron, and then your motor neuron says, okay, I have a signal for this muscle. It'll go through the ventral root, and it will go all the way to your muscle. And then you might move your hand or something like that. And in the middle here, you have what's called your mixed spinal nerve. So as you can see, in the dorsal root, you only have sensory fibers only. In the ventral root, you have only motor fibers. But in the middle, you have when it merges, you have your mixed spinal nerves. And then they will split off again into your ventral ramus and your dorsal ramus. So your ventral ramus, ventral meaning in front, so that will go to the front of your body. And it might go to your arms or something, it might go to your chest, but your dorsal ramus, dorsal meaning behind, that will go to your back. So yeah, that's just a basic overview. It can be quite confusing at the start, but you'll see this diagram more and more, and you'll become more familiar with it over time. And then you have different kinds of sensory fibers. So as you remember, I just mentioned myelin. So different nerve fibers will have different amounts of myelination. And so the myelination just depends on what type of neuron it is. So you have your A alpha, your A beta, and your A delta and C fibers. And I've listed them in order of most to least myelination. So depending on how much myelination they have, they'll carry different signals. So your A alpha, as you can see, the one on the left is quite fat. It will have the most myelination. It carries proprioceptive signals. So that's basically telling your brain where your hand is, where your arm is, where your leg is. It's basically telling your brain where your body is relative to the area around you. So A beta carries signals for touch, so anything you touch. A delta carries pain and temperature signals, and C carries pain, temperature, and itch. And then moving on to spinal tracts, which honestly were not very fun last year, but hopefully we can make it a little bit clearer. So in your spinal tracts, you have what are called first order neurons, second order neurons, and third order neurons. So your first order neurons are what feel the sensation. So they're on the most periphery. They might be in your fingers, your arms, etc. They carry the sensation from the peripheral sensory receptor to your spinal cord. So the cell body of the first order sensory neuron, it'll be located in the dorsal root ganglion. So it's just what we looked at before, the dorsal root ganglion here. And their receptor, it might be in a joint, a muscle, tendon, etc. It might be in your skin, wherever it is. So the first order neuron's job is to take the signal, the sensory signals from your hand to your spinal cord, something like that. And then the secondary order neurons, they are in your CNS. So this might be in your spinal cord, this might be in your brainstem. And basically the first order neuron synapses onto the second neuron, and it will take the signal up to your thalamus. which is a part of your brain. You don't need to know what the thalamus is. They kind of just throw a bunch of names at you, but just know it's a region of your brain. And then the third order neuron is synapsed by the second order neuron. And for the sense, it's basically for the sensation to reach our awareness. It takes the signal from the thalamus onto your primary sensory cortex. And that's really all you need to know. And then here are your spinal tracts, which were quite confusing the first time we saw them. And it might be a bit confusing for you now, but yeah, they basically vomit a bunch of information at you. But luckily, they're not extremely important. So just knowing the general details will suffice. So what spinal tracts are they're basically pathways of neurons to carry information from your periphery to your brain. And so what the dorsal column medial lemniscus pathway does, or this tract, is that it carries signals for highly localized fine touch. So that's really sensitive touch, like touch from your fingertips. It can also carry pressure, vibration, proprioception, and it will carry these through A beta and A alpha fibers, which are more myelinated. Excuse me. So what happens is the first order neuron, it enters ipsilaterally into the spinal cord. So ipsilaterally means on the same side. So if I have touch on my right hand, for example, the ipsilateral entrance, it means it enters the spinal cord on the right side. So then this first order neuron can travel up ipsilaterally. So it will stay on my right side, it will travel up the spinal cord on the right side. And then it synapses with a second order neuron in the nucleus gracilis and the nucleus cuneatus in the medulla oblongata. Once again, a bunch of names, you don't really need to know what they are. If you can commit them to memory, that'd be something a bit more extra. But basically what happens is it synapses with a second order neuron. This second order neuron decussates, which means it crosses over onto the other side of the body, onto the other side of the spinal cord. So it will go onto the left side of the spinal cord, basically, and it will travel up to the thalamus to synapse with the third order neuron. And then this third order neuron will carry signals from the thalamus to the somatosensory cortex. And then that's when you'll, realize, oh, you've touched this, or you feel the pressure, you feel the vibration, you feel the proprioception signals. And then another tract that you need to know is the spinal thalamic tract. So once again, just another pathway up from your periphery towards your brain. But this one carries a bit more of a different sensation. This one carries poorly localized crude touch and pressure, pain, and temperature through your thin A delta and C fibers. So they'll have less myelination. Once again, you'll have ipsilateral entrance. That just means, for example, if I had signals coming in from my left hand, that would enter the spinal cord on the left side. So ipsilateral means same side. And then, so the first one neuron enters on the left side. And then what happens though here, it will synapse immediately onto the second order neuron within the posterior gray horns. So that's the gray matter. Posterior just means behind. So the gray horns on the more behind part of the spinal cord. And then the second order neuron, which is an interneuron, it will decussate, it will cross over to the right side, and it will travel up contralaterally, meaning the opposite side, because you've crossed over, so it's contralateral now. And it will carry it up to the thalamus and synapse with the third order neuron. And then the third order neuron will carry the signals to and synapse in the primary sensory cortex again. So both of these tracks so far have been ascending tracks. So they carry information from your periphery up to your brain. Well, what descending tracks are is they carry information or signals from your brain to your body. So that might be telling your muscles to move, stuff like that. So you don't really need to know the descending tracks at all, except they exist and maybe know the name. So you have what is your pyramidal tract. So this consists of two individual tracts called the corticospinal, the corticobulbar tracts. And this basically goes down to your body, and it innervates the skeletal muscles all over your body, and it tells them what to do. And then you have your spinocerebellar tract, which is for proprioception. And that's just knowing where the body is in space. Once again, you don't really need to know these tracts. The two major tracts that you need to know are the ones we mentioned before, the spinal thalamic and the dorsal column medial lemniscus tracts. And yep, I think that's basically it. There's a table if you're more curious about descending tracts. Unfortunately, the stuff is kind of all vomited at you. It's a bit messy when you first learn it, but hopefully throughout the weeks, as you learn about it a bit more, it'll become a bit more familiar. So if you're feeling a little bit distressed or panicked right now, don't worry, that's perfectly normal. I remember looking at this last week and being like, what the heck, what is this? Like, they didn't really teach us any of this. They just kind of throw you in the deep end. But you'll be okay over a few weeks and it'll become more familiar the more you look at them. So don't worry too much, you've got time right now. And I think that's basically it. Yep, so does anyone have any questions?